Good morning, everybody. All right, so today we have some basics of DNA replication to go over. So let me get. Yeah, I need to see what I'm looking at on Craigslist. Here we go. That's more like it. All right, so we got up to here, right? Introducing this idea of a progressive sequence of events, right? And what we'll be doing, particularly on Friday and next Monday, is kind of adding information to these different points, right? We're going to be filling them out. We're going to be adding both what needs to happen and also the components that make that happen as well. So there's a lot of just terminology and names of things which you need to remember. So this is in many ways a more kind of concept and fact based section than before, which has been a lot more kind of math based, right? So we're moving away from that whole approach of, you know, probability and statistics and inheritance, and now we're actually looking at the actual molecular basis of a lot of those things. Hang on. Yeah, maybe. We'll see. It's more stuff to remember, and the concepts are kind of potentially trickier. Yeah, so that's funny actually when you say that, Amber. Some people have different, I think the best word is proclivities, right? So some people are better at math-based stuff that makes more sense to them. Other people are better at the kind of molecular biology aspects. It just kind of depends on your own personal strengths and weaknesses. But to be able to be good at all of it, you need to understand all of it. And so that's kind of one of the challenges of genetics. And it's also, in many ways, genetics and evolution are two courses in a series. It's almost like Gen Bio 1 and Gen Bio 2. And so a lot of the more population genetics and math-based evolution type stuff is in evolution, right? So Hardy-Weinberg equilibrium, for example, which if you thought any of the math stuff that we've done so far is tough, that's a lot kind of harder. And so traditionally genetics is taught with all of that, right? Going from molecular biology to inheritance to gene expression, population genetics, and then kind of a little bit of evolution maybe. But here we split that into two. And so most of the rest of this course is going to be more on the molecular genetics side of things. However, when you get to evolution, which is most likely the next course after this that you'll take, you might take microbiology perhaps, but at some point in the next semester or two, you'll be taking evolution. That picks up on the kind of bigger picture genetics, essentially. And so that's a lot of math and models and things like that. So you're not kind of getting away with it, unfortunately, right? So it's going to hit you at some point. But hopefully this will give you a good preparation for that. Also, as well, if there's stuff that you don't understand, just ask as well, right? I mean, that's kind of my job, really. It's not so much to teach you stuff, it's to help you understand things. Okay, so a bit of terminology, right? And there's going to be a lot of this. Where replication begins is called the replication origin, or in bacteria, if I can spell that right. Right, so let's see. Do we have that in another one? Yeah, there you go. So for example, this is the origin of replication in E. coli. It's called the ORIC. Let's get back to this. So there will be specific sites. In bacteria, there's typically just one, right? Because they have a circular chromosome. In eukaryotes, because we have long linear chromosomes, there are multiple origins of replication, right? It's not done in one go. It's done in lots and lots and lots and lots of goes, essentially. And also, each one of those, once replication begins, generates typically 2 replication forks. In bacteria and viruses, it may be just one. If DNA replication is unidirectional, in eukaryotes, it's always 2, right? So you'll see what we mean when we talk about replication forks. It's essentially the bit that kind of generates a fork, right? Where that DNA is getting opened up into single-stranded DNA and synthesized, right? So that's really what a replication fork is. We'll also be talking about replication bubbles. Essentially, that's where you have kind of DNA opened up and you have replication fork going that way and a replication fork going that way, right? So it kind of opens up into a bubble and then it just starts stretching out like that with new DNA being synthesized as it goes. Okay, the other kind of really key point to get is also that DNA replication is semi-conservative, right? And so what this really means is that you don't generate a new copy of DNA that has only new DNA in it or only old DNA, right? And the reason for that is each strand serves as a template for the synthesis of a new strand, right? So essentially this here is a perfect example, right? You have an old strand. This is called the parental strand or the template. And then you have the new strand called the daughter strand, right? Female terminology is used almost exclusively in biology, which is kind of interesting, right? Daughter cells, for example. So this new strand and this old strand make one new copy of DNA, right? Once this has gone on long enough, say in eukaryotes, this will become a chromatid, right? And this will become another chromatid. So these two will become sister chromatids, right? Not at this point because you haven't got far enough yet. But essentially, you will get 2 new copies or two new sets of double-stranded DNA, right? And each set will consist of an old strand and a new strand, a newly synthesized strand, right? And also, can't stress this enough, right? You're only ever going to add new nucleotides at the 3' end of the newly synthesizing strand, right? That's put on there by DNA polymerase, right? That's what essentially catalyzes. Let's go back up a wee bit. That's what catalyzes the formation of these phosphodiester bonds, right? So DNA polymerase is what generates that bond. Actually, it's this one here, the one at the top between the O and the P, And that's what sticks new nucleotides, right? These phosphate, sugar, nitrogenous base things to the newly growing strand, right? That's the role of DNA polymerase. And it can only add them to a 3' end because that's where the OH is, the hydroxyl group that is needed to add a new strand, right? You can't add a new strand, kind of butt weld it in a sense, to the 5' end. It has to go onto the 3' end. I can't stress that enough, right? That's why I keep talking about it. And that also causes a problem, which we'll get to later, right? So imagine essentially we're going to use the picture on the page, right? We've got a replication fork going up the screen, right? So this is the DNA is getting unzipped, going up, and the newly synthesized DNA has to be synthesized in that general direction, right? Both strands have to be synthesized at the same time. You can't do one and then come back later on and do the other one, right? You have to do them both at the same time. Their synthesis is actually linked, right, physically by the enzymes. But now you can see that we've got a problem, right? This daughter strand is all good, right? It's getting synthesized up the screen, right? Because its 3' end points up in this picture. This strand, however, is not. It is going in the opposite direction. That's the whole deal of the anti-parallel double helix, right? That's the problem that it causes. Has benefits too, but it also causes this problem. And so this strand is getting synthesized down the page, even though the whole replication fork is going up in the up direction. And so what we'll get into later is how that gets fixed, right? How do we deal with that problem, right? So that we can synthesize both strands at the same rate in the same direction, right? Anybody any questions? Okay. All right, so essentially we've got a bunch of things that we need to replicate DNA. Obviously, we need a template, right? And that template has to be single-stranded, right? DNA is a double helix, right? If I can try and do it with my fingers, right? It's double-stranded DNA. Those bases are going to be bonded to each other with hydrogen bonds. They're not available for use as a template. So that DNA has to be opened, right? And, you know, kind of made available so that those bases, right, are now free to pair with new nucleotides, right? That's what we need. That's how it can serve as a template. And so we have to have single-stranded DNA, which obviously leads the question, well, how is it opened, right? What do we unzip it with, right? Which we'll get to later. And so then we're going to be talking about, okay, well, you know, we have all these different tasks that we need to do. We're going to need lots of different enzymes to do them, right? And so these are the names of some of these. Some of these names kind of vary from prokaryotes to eukaryotes, right? So DNA gyrase, for example, is often called topoisomerase in eukaryotes. I'll just put that in the chat. And DNA gyrase is a type of topoisomerase, right? Which is part of the issue. And some of them are the same, like helicase and DNA polymerase are the same. In eukaryotes, DNA polymerase is numbered. In prokaryotes, they're numbered. In eukaryotes, they have Greek letters. Perhaps people who worked on eukaryote DNA replication were a little bit fancier. I don't know, like they learned Latin at school or something. And also you need a bunch of raw materials, right? You need these DNTPs, right? The A's, the T's, the C's, and the G's, right? And they contain both the bits that are going to get added and also the energy to catalyze that phosphodiester bond reaction. All right, so there are different ways in which you can copy DNA, and this really depends on the structure of the DNA, right? So bacteria have circular chromosomes, or actually have one circular chromosome, can't spell today. And they have things called plasmids, which are basically small circular chromosomes. And these are really interesting because they can carry things like virulence determinants. Oops, if I can spell that right. Antibiotic resistance. And these can also be shared between bacteria. Right, so one thing that's always a worry, you know, in terms of medicine, it's not just, you know, I talk a lot about people not completing a course of antibiotics and rise of antibiotic resistance, right? And so one worry is obviously that, you know, you'll select for bacteria that have mutations that allow them to resist or break down or be unaffected by antibiotics, right? That's kind of one mechanism. But also sometimes those genes which confer that resistance are on these little portable chromosomes called plasmids. And so if you're selecting for that, you can actually promote the transfer of that resistance from resistant bacteria to susceptible bacteria, right? It's called horizontal gene transfer. And so now you can actually, quicker than just selecting for the survivors, now you can actually confer resistance on large parts of the bacterial population, right? And that's the really scary part. And those can then kind of transfer it to others, and it can spread around as long as there's some kind of advantage to having that antibiotic resistance, right? That's kind of a big worry in livestock, where they just give them antibiotics just with their breakfast, basically. Also, things like that cause disease. So E. coli has a bunch of these. And so most E. coli is pretty benign. Some of it's absolutely terrifying. And a lot of the reason for that terrifyingness is encoded by these plasmids. And then you also have viruses. Viruses are weird, to be honest, and wonderful in some cases, not in our present day, obviously. But DNA viruses, actually, DNA viruses can have either circular or linear chromosomes. Not even really sure it should be called a chromosome, to be honest. DNA. I'm not sure there's really enough kind of stuff there to warrant calling it a chromosome. And then you have eukaryotes have linear chromosomes. Right, and so depending on what kind of DNA you're replicating gives you different ways of replicating that DNA, right? Because they have their own challenges, right? And so let's just do viruses first and plasmids. right? So this is called rolling circle replication. And the deal behind this is that it's very fast, right? And you can generate lots of copies of this DNA very, very quickly, right? So if you want to share it, for example, right, between bacteria, you need to make a lot of it so that you can give it to your buddies, basically, right? If you're a virus, the aim of the game is to basically replicate and generate more virus particles as quickly as possible for the cell dies or the host immune response, if it's in the host that has an immune response, kind of discovers you're there and kills the cell before you finish, right? So the aim of the game here is being fast. And the way that that's done is that we actually just replicate one strand at a time. That's kind of the exception to that rule that both strands have to be synthesized at the same time, right? Rolling circle replication kind of steps around that issue quite neatly. And what it does is it nicks, so it actually cuts, doesn't cut the DNA strand all the way through. So here's our double-stranded DNA, circular bit of DNA. What happens is you actually kind of cut one of those strands, right? And then you start essentially synthesizing that one new strand, and you go round the circle, right? That's why it's called rolling circle, right? And then when you get to the end, flink, off it trots, right? So now you have, and this can, as soon as you start one, right, you can basically kind of start again doing the other one, right? And so you generate lots of these linear copies, right? So they kind of flow around and they're going to get stuck back together. And now those circular single-stranded copies, these now serve as a template for synthesizing the other strand. So you can see while this is all going on, you're going to be circularizing the DNA that comes off first, right? You'll still be producing the linear DNA, but while you're doing that, that circular template can also be copied to generate this newly synthesized circular bit of DNA, right? And so this is very, very fast, right? Because of that reason why, you don't have to do both at the same time, right? So that kind of gets around some of the issues that we have with the whole anti-parallel nature of DNA. And it generates lots and lots of copies, right? So if you're a virus busy kind of replicating, and viruses have different modes of replication, but one of them is called a lysogenic mode, right? Which is basically when you generate as many bacterial viral particles as possible until the cell bursts, right? And then all those viruses go floating off and infect other cells and people and things, right? And so the idea is you got to do that real quick, right? Before that cell dies or before it's killed or before it's kind of chewed up by something, right? And this allows you to do that, right? So each one of these, if this is a virus, each one of these will become a new viral particle, kind of wrapped up in protein and sugars and stuff. If it's a plasmid, then again, it depends, if you're actively sharing this with other bacteria, you need a bunch of copies, right, of that. Or if you're actually being under selective pressure by antibiotic, right, and you need to fight off against it, then the more copies of that plasmid you have, more protein you can produce, the better you're able to neutralize or export or whatever that antibiotic, right? So now you can kind of understand where the merits of that is. Now, the thing that I've never really understood is why prokaryote, like bacterial genomes, aren't copied this way. That's a bit of a kind of an open question. I don't know if this potentially leads to more errors or what. I don't really know the answer to that question. Now, bacteria use what's called theta replication. Now, this can be, and this is really from I had a really neat GIF. I wish I'd put that in here. I'll have to go dig it out another time. This can be either uni or bidirectional, right? So we have, again, a replication origin or ORIC, right? Which we'll see later. And either both uni and bidirectional produce the same shape. They produce the same outcome, really. The difference is the speed. Basically, maybe this is what you do if you're replicating slowly in bacteria. This is what you do if you're replicating fast. Maybe it's down to what species of bacteria you are. I really don't know. I'm not much of a microbiologist, to be honest. Not at least bacteria. And so we have the same kind of idea. We open up the DNA, right? It's kind of twisted. And now we kind of open it up so that it's get my hands right. It's a bubble, right? And in unidirectional replication, we have a replication fork just going in one direction, right? So essentially it goes round the circle until it finishes back at the other end, right? So still we're getting semi-conservative DNA replication. We have each one of these strands serving as a template for the synthesis of new DNA. It's just half the speed, essentially. Whereas bidirectional replication, when you open this bubble up, you don't just have a replication fork going one way, you also have a replication fork going around the other way too, right? So it starts here and it kind of goes round until those replication forks meet, right, at the other side. All right, anybody have any questions? Nicht? Okay. So in eukaryotes, it's very different, right? So eukaryotes have huge linear chromosomes. Oh, I'm sorry. I keep feeling like it should be Friday, even though it's not. I thought it was Thursday yesterday. That was really sad. They have huge chromosomes, right? So just trying to think relative size. So bacterial genomes might be, in the order of a couple of 100,000 base pairs, give or take. Depends on the type of bacteria. Some of them, particularly pathogenic bacteria, are actually really small. Viruses can be as little as 5 to 10,000 to larger. Sometimes like trying to think. There's some that are really large. I'm trying to think of the disease that it causes. Can't remember the name of it. Basically, it makes you like really achy and tired long term. Can't remember the name of the disease, but anyway, in the high tens, essentially, of thousands of base pairs, right? And the reason, if you think about it, the reason why they have such small genomes is that A, they're not alive, right? So they don't need to encode genes that produce stuff they need for cellular respiration or for adapting to their environment, things like that. And also because a lot of the stuff which they need done to be able to replicate is done by the host, right? So viruses have real small genomes, right? Because they don't actually need very much information to get replicated. Bacteria have bigger genomes, right? In the hundreds of thousands of base pairs, perhaps, right? And again, they're also relatively simple in terms, they're not necessarily simple in terms of their behavior. Bacteria can do some really, really funky stuff. And eukaryotes, oh man, my spelling is terrible. That's supposed to be eukaryotes. Might have to go back and edit that in the chat. That's embarrassing. There you go. Not eukaryotes. You know, bacteria can talk to each other. They actually can behave as communities in lots of different ways, in terms of producing light or making biofilms, or, you know, they can act really effectively as communities. But still, they're unicellular organisms and they have relatively simple genomes. Eukaryotes can have crazy large genomes. The largest is wheat, from memory, which has, I think, in the order of 60 billion base pairs. Yeah, I think something like that. Can't remember the exact number. We have 3 billion base pairs in our genome, right? Which essentially we have 2 copies of that because we're diploid, right? So our chromosomes are huge. And so, A, they're linear, right? Not entirely sure why. Can't see why you can have really huge circular chromosomes, but anyway, they're not, they're linear. And also each chromosome is huge, right? And so there's no way you can start, as you do with bacteria, with just one origin of replication and even bidirectionally replicate that chromosome in any kind of reasonable time frame, right? And one of the reasons why eukaryotic or like human cell division takes so long compared to bacteria, some bacteria can divide in 20 minutes, for example. Mouse cells and human cells take about a day to be able to divide, right? And most of the reason for that extra time is the time taken to copy the DNA, right? And so in eukaryotes, what we have are these multiple replication bubbles, right? So we have a replication origin, and you can have hundreds of these, right? Potentially, all dotted along the chromosome. They'll also be characterized by a high percentage of A's and T's as well, right? Because we want to be able to kind of, let's see how, kind of open up, can't do the hand thing. Basically, you twist the DNA, there you go, like that, or something like that, to pop open the strands, and you have multiple sites of this all the way along the chromosome. And so what happens is, obviously, each one doesn't necessarily start at the same time, and you'll have your bidirectional replication going along, and essentially it'll keep doing that until it either hits the end of the chromosome or it hits another replication fork or replication bubble, right? And these will then fuse, right? You'll fix the gap kind of as a part of the DNA replication machinery. Oh, no, who keeps calling me? Anyway, kind of weird to be having my phone ringing in my office. It's in a little time I spent here. Okay, so let's look at that in a little bit more detail, right? And so this whole bidirectional nature becomes an issue with any organism that replicates both strands at the same time, right? So here, for example, right, we have this is rolling circle replication, right? We're starting to replicate our new strand. This outer one kind of gets wound off, right? And that then forms a new template for replication, right? If you remember from up here, right? So that original template kind of gets linearized, turns back into a circle, and then gets copied while the other new strand is being done. right? So this is unidirectional and one strand at the same time, right? So there's no issue here, right? Because we only have one strand going synthesized in a 3' direction, right? When this one gets picked off and circularized, we'll also have one strand also going in the 3' direction, right? So the strand which is synthesized in the direction of that replication fork movement is always called the leading strand. Let's type that up. So that's simple, nice and easy. However, when you have both strands being synthesized at the same time, you have this issue. What's in here? That's very interesting. Okay, so again, we have this movement. Now we're going from right to left across the page. We have our replication fork moving from right to left. And so we have one DNA strand, right, which goes 5' on the left to 3' on the right. Because it's anti-parallel, the new strand is going to be synthesized in the opposite direction, right? That direction happens to be towards the direction of the replication fork movement, right? So again, we now have a leading strand kind of going in the same direction as the replication fork. Now, the other strand needs to be synthesized in the opposite direction, right? But these two strands, they're linked, right? You can't synthesize one and then the other like you do in rolling circle replication. You have to synthesize both at the same time. And so this then means that the lagging strand, right, the strand which needs to be synthesized in the opposite direction, gets done in small chunks, right? Basically by flipping the DNA back over and synthesizing it backwards, right? So we have these small little chunks, and they're small chunks because you essentially start ahead, synthesize backward until you hit what you've already made, right? And these are called Okazaki fragments, right? So I'm gonna move this over a little bit. So it's kind of hard to describe the lagging strand because it is still actually synthesized in the same direction as the replication fork, but the only way it can be done that way is by synthesizing in small chunks. Right, this is the tricky thing to get your head around, right? Oops. No, I don't want to do that. Don't do that either. Go away. Here we go. This template strand here, right, which is going in the wrong direction, right? So this template strand has a 5' end here, and so the new strand is synthesized in the other direction from 5' to 3', right? And so basically that strand is kind of looped over and bent backwards, right? So you can still synthesize it, the new strand, 5', 3', in the same direction, but you can only do a little bit, and then you have to flip it back in again, synthesize a little bit, flip it back in, synthesize a little bit, flip it back in, and keep doing that in little chunks. And this is called the lagging strand, not because it's slower, like the DNA gets synthesized slower. It's called the lagging strand because it takes time to fix these gaps, right? So both strands are at the same rate. They're synthesized at the same rate, but it takes longer to essentially fix the gaps between the Okazaki fragments. That's why it's called a lagging strand, not because it takes less time to synthesize it, but because it takes less time to fix all this. If you think about the leading strand, right, you get one leading strand per replication fork, right? So if you have a large bubble, right, essentially you'll have, this will just be all one strand, right? This can go for potentially thousands of base pairs, right? And so all you need to do is kind of synthesize fix the gap where different forks meet, right? When those leading strands kind of, butt into something that's already been done. With the lagging strand, you have hundreds of those fragments that you have to fix, right? Because they're really pretty small. Even in bacteria, they're still, in the thousands of base range. In eukaryotic cells, they can be in the 1 to 200 base pair range, right? So if you think about a bacterium that has a genome, let's say unidirectional, that's say 200,000 base pairs, 200 kilobases, is going to have one 200 kilobase leading strand and potentially 200 Okazaki fragments, right? And so it just takes time to fix those. Anybody have any questions about Okazaki fragments? This should be, by and large, a recap of GenBio 1, but that may have been some time ago. Okay, and so when you look at it in greater detail, right, in each individual context, right, this is bidirectional theta replication, so this would be in bacteria. And so you're going to have one origin of replication, and from each origin, you're going to have a leading strand, right, going in the 5 to 3' direction, and you're going to have a lagging strand full of Okazaki fragments. Same on the other side, right? It's going to be on the other strand. You'll have a leading strand and a lagging strand, right? In eukaryotes, you have the same, right? But the difference here is that you're going to have over there and over there, you're going to have other origins of replication and other replication bubbles, which will eventually they'll merge into each other. And once you've done all of them, obviously, then you have your two fully synthesized strands of DNA, your sister chromatids. All right, questions? If not, we'll move on to the next one. There are some really good videos on how the lagging strand is synthesized that would be worth watching. It's a little bit easier to see visually rather than to describe in words. Okay, so we're just going to finish up the rest of the class, starting DNA replication in prokaryotes. And the reason why we talk about prokaryotes is because they're a little bit simpler and have, by and large, the same mechanism and components. They just have a few of them. All right, so again, we have the same idea of phases, right? First, we need to start the process, right? We need to find that origin of replication. We need to open the the DNA, or actually bending the DNA, right, which pops it open. We need to unwind it and do that as a dynamic process, right? As we're moving along, we need to elongate, and then at some point we have to stop. So essentially, to initiate DNA replication in E. coli, you need a bunch of different proteins. So the first is you have these initiator proteins, right? What they do is they recognize this DNA sequence, right? There are a lot of proteins that are DNA binding, right? So remember we talked about the ability of DNA to recognize, ability of proteins to recognize DNA, both its shape and its sequence. That's what you're seeing here, right? So this origin of replication has a particular DNA sequence, and that is recognized by these initiator proteins. And what they do is they bind that origin of replication and they bend the DNA, right? So essentially they're going to be kind of twisting the DNA around, and in doing so, it kind of pops open a bubble. It's almost like, you know, you get a piece of rope, and if you kind of twist it in a certain way and then kind of give it a, you'll force the strands to unwind, right? You put in kind of motion which is opposite to the twist of that rope. And that's what these initiator proteins do, right? They don't do anything other than that, right? Their job is simply to recognize that origin of replication. These are going to be produced at a certain point in the cell cycle, right, of the bacteria. And that then kicks off the whole process, right? So you get this little bubble of single-stranded DNA, right? Once that single-stranded DNA is opened, then you have, rather unimaginatively named, single-stranded binding proteins. These do not play a role in DNA replication itself. What they do is they hold that bubble, that single-stranded DNA open, and stops it from just sticking back to itself, right, and forming double-stranded DNA again. And in doing so, they allow the entry of this protein here and here. This is called helicase. Helicase is an unwinder. Right? Its job is to move along, process along the DNA, right? So it kind of like walks along and act like a bit of a plow or a wedge, right? And so its job is to kind of force its way between the two strands, forcing them open, right? Causing them to unwind and open up. Now, has anybody tried like sticking a pencil or a pen into a rope and pushing it along the rope? Say you want to kind of get the individual strands or something. What happens when you stick a pen in a rope and you kind of push it along? What happens to the rest of the rope? Has anybody ever tried that? You should try it. It's fun. You haven't lived, Sophia. Everybody has to unwind a piece of rope once in their lives. Right. What happens is, as you're unwinding it, you're causing the rope to rotate, right? Because if you reduce the twist in one part, you induce more twist in another part, right? And what that does is if you kind of stick that bit of rope to something, or it's a circular piece of rope, it will kind of coil up further and further and further and further until it gets into like this kind of tangled bunch, right? Almost like kind of detangling Christmas tree lights, stuff like that, which is not my favorite job. And so that's called supercoiling. And if that continues, essentially what you'll get is just like this tangled mess of coiled DNA. And so that's what DNA gyrase does, is it reduces that supercoiling, right? And we'll talk about that in more depth on Friday. I think that's probably enough for today. All right. Does anybody have any questions before we call it a day? Awesome. Okay. Well, thank you for your time, everybody. And it's still not the weekend, unfortunately, but it's closer, halfway. And I will see you either in lab this afternoon or on Friday when we'll continue bacterial DNA replication. All right. Stay safe, everybody.